Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 35. The topic is the synthesis and breakdown of polymers. Discussing agenda for the topic will be polymerization and hydrolysis. Lecture outcome. After watching and listening this lecture, students will know about the biological macromolecules and how they are assembled into polymers and disassembled into monomers. Introduction. Macromolecules are polymers built from monomers. Over the last few lectures, we have discussed different types of biological macromolecules and you people studied different types of monomers which combine and form large molecules called polymers. Large carbohydrate, proteins and nucleic acids, also known as macromolecules for their huge size, are chain-like molecules called polymers. The Greek word polys means many and meros means part. So polymer is formed as a result of the combination of identical or nearly identical building blocks called monomers. For example, in the case of carbohydrates, we have simple sugars as monomers. In the case of DNA, we have nucleotides as monomers. In the case of proteins, we have amino acids as monomers. So these monomers combine and they form large molecules called polymers. So how we define a polymer? A polymer is a long molecule consisting of many similar or identical building blocks linked by covalent bonds, such as a train consists of a chain of boxcars. You may have seen a train which transport goods and this train have lot of box-like structures in which the goods are transported. So the polymer also somewhat looks like a box train with attached boxcars, means it is made up of identical repeating units. The repeating units that serve as the building blocks of a polymer are smaller molecules called monomers. So monomers are the basic building blocks of the polymers. The Greek word monos means single. In addition to forming polymers, some monomers have functions of their own. Some monomers perform function on their own, whereas the monomers can also aggregate in the form of polymers. For example, glucose is a monomer and it can directly be used to derive energy when it is incorporated into the glycolysis, and in other cases the glucose monomers combine and they either form the starch or cellulose. Although each class of polymer is made up of a different type of monomers, the chemical mechanism by which cells make polymers or polymerization and breakdown are similar for all classes of large biological molecules. Those four different types of macromolecules, each of them have characteristic properties and functions, but the basic philosophy behind their synthesis and breakdown is same. The chemical reactions involved in the synthesis and breakdown of these macromolecules or polymers is same. In cells, these processes are facilitated by enzymes. Enzymes are proteins in nature. Majority of the enzymes are proteins in nature. Sometimes RNA also act as an enzyme, which is called ribozyme. So RNA is not an enzyme, but predominantly RNA acts like an enzyme, but it is not protein. So all enzymes are not proteins. Here what we are referring to the enzymes is the fact that they catalyze the synthesis of polymers. The reaction that connects a monomer to another monomer or a polymer is called a condensation reaction. So when two monomers combine, the reaction which takes place is a condensation reaction or a dehydration reaction. What happens in that reaction? In that reaction, two molecules are covalently bonded to each other with the loss of a small molecule. So generally a water molecule is removed during the condensation process. If a water molecule is lost, it is known as dehydration reaction. Generally, the formation of polymers in the case of carbohydrates and proteins are dehydration reactions where a water molecule is removed. For example, carbohydrates and protein polymers are synthesized by dehydration reaction. Now each reactant contributes part of the water molecule that is released during the reaction. Water molecule of course consists of a hydrogen ion and a hydroxyl ion. So when two monomers combine and form a polymer, one of the monomers contributes the hydrogen ion and the other monomer contributes the hydroxyl ion. So a bond is formed and water molecule is released. One provides hydroxyl ion while the other provides the hydrogen. This reaction is repeated as monomers are added to the chain one by one, lengthening the polymer. So these reactions are repeated again and again as the monomers combine. Here is you can see a graphical representation of the formation of a polymer. Those are monomers. They combine with the help of a dehydration reaction. One of the monomer is contributing hydrogen ion, the other monomer is contributing the hydroxyl ion. The water molecule is released and the bond is formed. This bond is a glycosidic bond in case of carbohydrates, and it is a peptide bond in case of proteins. So this reaction is repeated again and again, and a long chain is synthesized, which is called polymer. Polymers are disassembled to monomers by hydrolysis. Now, if a polymer is intended to be broken down into its individual building blocks, into its individual units called monomers, then the reaction is exactly the opposite one. Decondensation reaction prepares polymers, and hydrolysis reaction, it results in the breakdown of polymers into monomers. And this is essentially the reverse of the dehydration reaction. In dehydration reaction, polymers are formed from monomers. In hydrolysis, monomers are formed from polymers. Hydrolysis means water breakage from the Greek word hydro means water and lysis means break. The bond between monomers is broken by the addition of a water molecule with a hydrogen from water attaching to one monomer and the hydroxyl group attaching to the other. So you can see the hydrolysis process is the exact opposite of the dehydration process. In dehydration, one of the monomers contribute hydrogen, the other monomer contribute hydroxyl, and water molecule is eliminated. In dehydration reaction, the exact opposite reaction occurs, and this in hydrolysis exact opposite reaction happens, where water molecules is dissociated into hydrogen and hydroxyl, and to one of the monomer, hydrogen is donated into another monomer, the hydroxyl ion is donated, and the bond is broken down. An example of hydrolysis within our bodies is the process of digestion, the breakdown of food materials. Here you can see a representation of the process of hydrolysis, in which the polymers are broken down into monomers. You can see this process occurs in the presence of water. A water molecule breaks down and hydrogen ion is given to one monomer, hydroxyl to another monomer. The bulk of the organic material in our food is in the form of polymers that are much too large to enter our cells. Now the organic molecules are present in our food, and they are in polymers. They are broken down into their individual building blocks or monomers. For example, complex carbohydrates are broken down into simple sugars, and proteins are broken down into amino acids. Within the digestive tract, various enzymes attack the polymers, speeding up hydrolysis. Released monomers are then absorbed into the bloodstream for distribution to all body cells. Those cells can then use dehydration reaction to assemble the monomers with new different polymers that can perform specific function required by the cell. So in food we consume monopolymers which are in complex form, and inside our body during the process of digestion, these complex polymers are broken down into their individual units or monomers, and then these monomers or these building blocks are used by our body to synthesize polymers of other types. For example, you consume proteins from the outside. These proteins are broken down into amino acids, and then these amino acids are incorporated into proteins again. Which type of proteins are formed? The proteins, the instruction of which are there in our DNA. So we basically consume food to derive these basic building blocks. A dehydration reaction and hydrolysis can also be involved in the formation and breakdown of molecules that are not polymers, such as lipids. Lipids are not true polymers because there is so much diversity in their structures. The only thing which classify them together is their characteristics of hydrophobicity. A cell has thousands of different macromolecules. The collection varies from one type of cell to another. The inherited difference between close relatives such as humans, siblings, reflect small variations in polymers, particularly DNA and protein. So there are phenotypic differences between organisms, and this is because of minor changes in on the DNA level. These minor changes are then expressed in the proteins, and then proteins in turn control the phenotypes. Molecular differences between unrelated individuals are more extensive, and those between species greater still. So the molecular differences increases when the phylogenetic or the evolutionary relationship between the two species increases. Suppose two individuals have a recent common ancestor, then they will have more similarities with one another, and if they have a distant common ancestor, they will have more differences on both the phenotype level and molecular level. The diversity of macromolecules in the living world is vast, and possibly variety is effectively limitless. What is the basis for such diversity in life's polymers? These molecules are constructed from only 40 to 50 common monomers and some others that occur rarely. So building a huge variety of polymers from such a limited number of monomers is analogous to constructing hundreds of thousands of words from only 26 letters of the alphabet. So suppose there are only 20 different types of amino acids, and there are hundreds of thousands of different types of proteins built up of only those 20 types of amino acids, and similarly we have so many variations in the genetic material in DNA, although it is only made up of 4 different types of nitrogenous bases. So what is the basis for these differences? It is the sequence and the arrangement of the nucleotides or the amino acids or the sugars which help in the formation of different polymers. The key is the particular linear sequence that the units follow. However, this analogy fails far short of describing the great diversity of macromolecules, because most biological polymers have many more monomers than the number of letters in even the longest word. So some of the macromolecules have lot of monomers instead of only 4 in proteins, and instead of only 20 in proteins and 4 in nucleic acid. Proteins, for example, are built from 20 kinds of amino acids arranged in chains that are typically hundreds of amino acids long. So here is a summary of the 4 types of biological macromolecules which we have studied over the past few lectures. Carbohydrates, they have types monosaccharide, disaccharide and polysaccharide. Their function is immediate energy and stored energy and structural molecules. Lipids are made up of fats, oils, phospholipids and steroids. Their function is long-term energy storage and membrane components. Similarly, proteins have lot of functions which we dedicated a whole lecture to, and their function is support, metabolic, transport, regulation and motion. Nucleic acid consists of DNA and RNA, and the function of nucleic acid is the storage of genetic information. This is the end of the lecture. Thank you. Allah Hafiz.